So I will continue with the second lecture that is on the program, and which will deal with the rather quality aspects and other requirements to get your biosimilar approved. So this is an overview of the guidelines of from the European Medicines Agency to get guidelines for the pharmaceutical industry to which they have to adhere or take into account when developing a biosimilar. We have an overall guideline. Then it's important to realize that there is a general guideline on the quality evaluation of your biosimilar. There's also some general guidelines on the non-clinical and clinical evaluation of your biosimilar relative to the reference product. And what is also important, and a little bit of a difference between Europe and the US FDA, for instance, is the fact that in Europe there are also some product-specific guidelines with respect to the non-clinical and clinical evaluation of the different molecules. And that's because you may understand that when we look at a simple molecule like erythropoietin, we may have to look at it in a different way compared to, for instance, a complex molecule such as a monoclonal antibody, because the relationship between the structure and the function for erythropoietin is much more simple than for a monoclonal antibody. And these guidelines will also take that into account. And if we look at a monoclonal antibody, for instance, the guideline will ask or will tell the companies that they have to look, first of all, that the binding of your antibody, the biosimilar antibody, the binding to the target has to be compared with the binding of the reference product to the target. But as you may know, the functional properties of an antibody do not only depend, in many cases, do not only depend on the interaction between the variable domains that are indicated here on top of this structure and the target, but will also depend on the constant region and the physicochemical properties of the constant region. And this constant region, the Fc part, is responsible for binding to quite a number of different sets of Fc receptors that do occur on the cell. So you will also have to demonstrate that your biosimilar infliximab, for instance, does not only bind to the target, TNF, but you will also have to demonstrate that your infliximab biosimilar binds in a very similar way to the different Fc receptors, in particular in vitro cellular models. But obviously, you want your product not only to bind to the target, you will also have to demonstrate that subsequent to the binding of your biosimilar to the target, this biosimilar induces the same effect that you expect. For instance, in the situation of infliximab, it binds to TNF-alpha, but it also has to neutralize the activity of TNF-alpha. So you will also have to demonstrate that your biosimilar copy, apart from binding to the target, also, for instance, neutralizes the target in the same way and to the same extent as the reference product does. And obviously, the same for the Fc-mediated functional effects. You will also have to demonstrate that your copy, your biosimilar, induces the same kind of ADCC, CDC, and so on upon binding to these Fc receptors. So it's not just looking at binding, it's looking at binding to the different regions and actually the different functional consequences upon this binding. So this makes life for a monoclonal antibody biosimilar a little bit more complicated than, for instance, for erythropoietin. That's also something one has to take into account. Now, if we look very briefly to the registration requirements of a medicine, this is for an original product. You obviously have a full quality, a full non-clinical, and a full clinical dossier that has to be submitted. That seems obvious to all of us. For a biosimilar, as indicated, we want to make some shortcuts, but some shortcuts that are that can be explained, of course. One important point is that when you have an approved biosimilar, you should realize that it has been approved based on a full quality dossier. There are no shortcuts with respect to the quality aspects of your product. And in addition, as you know, you also have to provide an entire set of data that demonstrate that the biosimilar is indeed sufficiently comparable to the reference product. And once that is the case, as I already indicated, the amount of non-clinical data that you will have to provide will be much less, because we have extensive experience with the reference product. And if the reference product, for instance, is expected or has no off-target side effects, for instance, we may expect it to be the same for the biosimilar, on the condition that we have sufficient comparability data at the quality level. So what stays in dark blue is something that has to be included in your biosimilar registration file. What is in gray is usually not needed anymore. And what it may be important, and we will come back to that later, that if we look at the clinical part, you may notice that it may be sufficient to demonstrate biosimilarity at the clinical level in one indication for which the reference product has been authorized, and for the biosimilar you may get approval for all the different indications for which the reference product has been granted an authorization. This is the concept of extrapolation, which will also be discussed later. It also has an important implication for the use of a biosimilar. The fact that we have, first of all, less clinical data, not only in less indications, but also usually in a smaller number of patients, implies that we will need some more post-marketing information once a product has been approved. So we may feel the need to gather more safety data in larger populations, in other disease indications, have more confirmation of the efficacy. And what is also very much important, on the long term, larger populations, we may have to confirm the real comparable immunogenicity, which is usually impossible to identify or to detect within a short term in small study populations. But again, this will be discussed later. So where are we in Europe at this moment? Just a brief overview. There are a few products that have been refused or have been withdrawn, all for the same reason, because there was something, let's say, wrong with the quality evaluation of the product. Differences that were not acceptable, differences that were too large. In some cases, differences with respect to the efficacy of the product, and so on. Just to illustrate that this process is pretty stringent, and you need to adhere to the guidelines and have a good quality product before it can be approved as a biosimilar. And you may notice that even a relatively simple molecule such as filgrastim, here the pegylated form, there are actually a few also recently that have not been approved yet at this moment. Here's some details why they have been produced. For instance, insufficient details on the entire process, which just means that we cannot trace back the quality of the entire process, and so on. Insufficient quality, for instance, with respect to comparability. Here we have some differences, important differences with respect to impurity, for instance, a lower efficacy for this proposed biosimilar of interferon alpha-2A, which also demonstrate that they look at all the different aspects. There are approximately 41 dossiers that have been approved as of today, basically within the Europe or by the European Medicines Agency. What may be important to notice is that you may see that for quite a number of products, for instance, for filgrastim, we have 4 that were approved in 2008, another 2 in 2009, 1 extra in 2010, for instance. Another one in 2014, which illustrates that we will have to deal in real life with different biosimilars from different sources. And this is also something that will have an impact on the floor, as we will come later in this morning session. Another important point is that may make life a little bit more difficult. That is, if we have, for instance, here 4 times filgrastim, and we have here 6 rituximab. This does not mean that there are, for instance, for the rituximab, 6 different sources. You know, and I'm not an expert on commercialization of pharmaceutical products, but sometimes a company introduces 4 times the same dossier to have it marketed under different trade names, but it's actually the same product, just with another label on it. For instance, for the 6 rituximab, these are actually 4 products that are exactly the same. These are all biosimilars, but the same biosimilar from the same manufacturer, basically, but will be marketed under 4 different trade names. And the other 2 are also, again, from another manufacturer, but again, 2 identical process, which again will lead to some complications when implementing biosimilars and when discussing switching or interchangeability issues under certain conditions. Just to illustrate to you that we have quite some experience within Europe with biosimilars, but also to illustrate that there will be some complex issues arising when implementing them. It's still not finished yet. We have approximately 13 files that are currently under review. What kind of tests do have to be submitted for your registration? And you see here a few. We will not discuss all of those. It's just to illustrate you some physicochemical analysis. And you see here N-terminal sequencing, C-terminal sequencing, disulfide bonds, circular dichorism to get some idea of the secondary structure. Some size exclusion chromatography to check the number or the extent of aggregation of your protein, which is also an important parameter. Some isoelectric focusing. Glycosylation, as you can see here, and glycosylation is also a complex procedure and will require different analytical approaches to evaluate the type of glycosylation, the extent of glycosylation, the position of the glycosylation, and so on, all to demonstrate that your biosimilar is glycosylated to a very similar way as the glycosylation of the reference product. And yeah, I mentioned this already, in particular for monoclonal antibodies. This is a bit more complex. I will not repeat this now. Yeah, this is just again taken from the European Public Assessment Report for the first infliximab monoclonal antibody biosimilar, where they described the specific FAB-related evaluation that was done. As I mentioned here, the TNF binding SF, for instance, TNF, not only the binding, but also the neutralization has been demonstrated for this particular process. Then actually some functional in vitro cellular models to demonstrate the impact of the binding of the biosimilar to TNF-alpha. And also, as I indicated, the FC-mediated effects on particular cellular systems. And in addition, the company that submitted the biosimilar dossier for the first infliximab biosimilar also actually did some detailed 3-dimensional analysis, X-ray structure, X-ray crystallography, to demonstrate the real 3-dimensional structure of the molecule. If we look a bit more in detail for these different European public assessment reports, and you know that if you look at the website of the European Medicines Agency, for each approved medicinal product, you can find a summary of the all the properties that have been taken into account to approve the molecule. It's obviously not the 100,000 pages dossier that has been submitted. I mean, it does not contain any confidential information, but for instance, for the biosimilar, you will find in many European public assessment reports the basic information on which the CHMP, the scientific committee, has evaluated the biosimilar relative to the reference product. And there you can find, for instance, or you will notice that they indeed reported differences at particular properties, differences at the level of glycosylation, for instance, differences at the level of oxidation, as we can see here, differences in impurity profile, and so on. For the biosimilar infliximab, for instance, they also noted differences in the extent of afucosylation. And without going into too much molecular details, the extent of afucosylation has a clear impact, this is very well known, on the binding of the Fc part to particular Fc receptors, and this may have also an impact on the functional properties of your monoclonal antibody in vitro, but ultimately may also have an impact on the overall in vivo efficacy of your molecule. So if you find a difference with respect to this particular quality attribute, this will be what we call a critical quality attribute, because differences in this particular quality attribute may have an impact on the clinical properties of the molecule, for instance. So this just demonstrates, if you look at the European Public Assessment Report, that what I told you this morning, that indeed it will be impossible to make an exact identical copy of a reference product, but you may be able to make a copy that is highly similar to the reference product, but never fully identical to it. And to give you some examples of differences that have been reported, and some of them may lead to some discussion or to some ambiguous interpretation. And as I mentioned already, this lower degree of afucosylation, a lower binding affinity, and they actually also demonstrated lower affinity, lower activity within a ADCC assay, a cellular in vitro assay, to evaluate the extent of antibody-dependent cellular cytotoxicity. Obviously, it's important that when I highlight some differences, this is not the only property, of course, of this biosimilar. You have to look at it in its the entire collection of all the different properties that have been reported and compared to the properties of the reference product. So ultimately, the experts decided that these differences in vitro, from a physicochemical point of view, and from the in vitro functional properties in view, actually it was not to be extrapolated to actually the expected clinical impact that it could have. So these differences are considered to be non-clinically, not relevant within the clinical context. We may skip this one. This one is a very important case, which illustrates that the evaluation of a biosimilar is not always a black and white situation. So what do we find in the European Public Assessment Report when they describe the approval process and the properties of a biosimilar SB2 versus the reference product of infliximab? One of the typical properties that is associated with infliximab, it's a chimeric molecule, chimeric monoclonal antibody, and all the patients, not all the patients, I'm sorry, but within a period of 6 months, let's say that on average approximately 40% of the patients will develop antibodies against the drug. This is something typical we know for the reference product. So we're obviously not surprised that also for the biosimilar, a number of patients will develop antibodies. So this obviously has to be evaluated during the clinical development of the biosimilar, and they figured out that, and ADA stands for anti-drug antibodies. So this refers to the immunogenicity of the drug. And the patients treated with a biosimilar, there were 47% that had developed antibodies against the biosimilar infliximab versus only 38% antibody development to the reference infliximab molecule. And obviously when you develop antibodies against the drug, this may have an impact, and that has also been illustrated in some cases, this may have an impact on the efficacy of the product. If you have neutralizing antibodies, then obviously your drug will not work anymore or work much less. And if they looked at all the data that were present, for some of the experts, the CHMP is the assembly of scientific experts that do evaluate the quality of the dossier in all its aspects. And you will notice that actually 14 out of 36 of the scientific experts were not convinced that this difference would not have any impact on the clinical efficacy on the long term for this product. But, you know, in a democracy, it's the majority that decides. So there was, if you calculate this, you obviously have seen this, the majority was indeed in favor of the approval, and the product was indeed then approved. I'm not saying that I conclude that this biosimilar is not a good biosimilar. It just illustrates that even the scientists who had information, much more information that we have, were not entirely agreed on whether it should be considered as a true biosimilar or not. Another point is, when you evaluate the clinical biosimilarity, as I have briefly mentioned, you don't have to have a full clinical package. So it's also very important that you select a particular patient population that is indeed representative for the other patient population. And you will also know that depending on the indication, depending on the patient population, you may have also concomitant adjuvant treatments, for instance, such as methotrexate. Methotrexate will also have an immunosuppressive effect. So these data, 47% immunogenicity versus 38% immunogenicity, was also obtained in a patient population that was treated with methotrexate, that has even an immunosuppressive effect. So this actually means that you have no idea whether this difference would even be higher in the absence of methotrexate, for which some indications were approved. So this also raises some, let's say, uncertainty. And for the sake of time, I will skip this one. Again, some differences that were observed, but not assumed not to be of clinical relevance. But you see here again differences in afucosylation and so on. It also depends whether particular quality attributes are assumed to have an impact on the mechanism of action, yes or no. So it's all on a case-to-case basis. What we will require for infliximab may not be the case for adalimumab, for instance, and vice versa. Now, another important example, again extracted from the European Public Assessment Report, is the, let's say, the way the scientific committee deals with the data that have been provided. So, for an etanercept biosimilar, as before, they have found that a significantly lower incidence of immunogenicity was observed for the biosimilar relative to the reference product. 1% for the biosimilar versus 13% for the reference product. We can say that's very good, because it gives you the impression that the biosimilar, in terms of immunogenicity, is better than the reference product. But obviously, we want to have something that is similar, not something that is better. So that's also very important in the biosimilar concept. You have to demonstrate that your product is similar. Definitely not worse, but also not better, because if you prove that it's better, then you're not sure whether you're really working with a similar product, and then the whole concept may not be able to be applied. So 1% versus 13% is obviously a little bit of a problem in terms of regulatory point of view within the biosimilar concept. You may also know, and I won't go into detail again, measuring anti-drug antibody response is also a technological challenge. It also depends on the patient sample, it depends on the drug concentration, and so on. So within the entire dossier, it was decided that for certain time points, week 4 and week 8, probably the data on the anti-drug antibody response were not fully reliable for whatever reason. Okay, yeah. So what they did is they decided that they should reanalyze the data by removing the data from week 4 and week 8. And then they came up with 0.3% versus 0.7%. Instead of 1% to 0.3, and the 13% decreased to 0.7%. Same data, but just removing week 4 and week 8. Obviously, you will realize as a scientist, if I would like to publish data and I will have to tell the editor that I deleted some data to have a more better outcome, this paper will never get accepted. So again, I'm not saying that there is something wrong with the biosimilar, I'm just saying that, from a scientific point of view, there are some doubts about how these data are being looked at. And ultimately, it's concluded that it's premature to conclude anything about the immunogenicity. So you see that there are some data that are not fully, and I will skip this one. I'm almost at the end. Terminology is important because, and definitely also I think in this region, if we talk about biosimilar, you should be sure that we are talking about a true biosimilar, which means that it is a very good evaluated version relative to a reference product that has already been authorized, for which the similarity has been demonstrated. And we may only detect only small differences. I won't deal too much with second generation, but you know that, for instance, for erythropoietin, we have not only erythropoietin, but also darbepoietin, the longer acting version. So this is obviously not a biosimilar of erythropoietin. This is a second generation or a next generation version. Filgrastim versus pegfilgrastim, for instance. But apart from that, you may also, and I'm sure you're aware of that, they're also circulating some copies of products, a whole set of erythropoietin drugs that have never been compared to any existing authorized drug. So these are usually referred to non-comparable biologics, which means that they have been produced by somebody in some countries, but they have never been extensively characterized compared to reference products. So these are definitely not falling under the category of biosimilars as we discussed right now. And these are just some examples which I can skip. And I won't go into too much detail, but it's definitely such that these non-comparable biologics have never been demonstrated to be biosimilar, and therefore this is a small slide. If we look here very quickly, this is a biosimilar of Eprex, for which you see an isoelectric focusing. This pattern illustrates the different glycosylation forms very briefly, and you see that these different patterns, the approved biosimilar and the reference product, is very, very similar. You can see this very quickly. If you look at the whole set of other erythropoietin copies that have been introduced somewhere in the world, but definitely not in Europe, not in the US, not in Canada, and so on, you will see that the pattern of glycosylation is very much different between those different copies, also illustrating that definitely they are not to be considered as biosimilar. And, you know, these are a few Dr. Reddy's products, for instance, Reditux. Rituximab is a product that is produced in India, but has never been compared to any approved product within Europe or within the US. You will also see here a 17-patient phase 2 study. This is a very limited number of data, obviously, and also in China, for instance, some copies that are not to be considered as biosimilar. So in conclusion, if we're talking about biologicals, in general, we should realize that these are complex molecules, often composed of different domains that are important for the function. We are not dealing with one particular variant, but with heterogeneous protein population. The entire process will determine the ultimate properties of the molecule. We should not consider them as interchangeable, and we will come to that after the coffee break. And obviously, we need quite some post-marketing surveillance. With respect to the experience that we have so far in Europe with the approved biosimilars, is that first of all, the process is very well challenging and very rigorous, controlled. The pharmaceutical quality is guaranteed. That's per definition. But we notice that there are definitely, as we could expect, differences in quality attributes between the reference and the biosimilar. And the actual challenge is to demonstrate that any of these differences have no clinical impact on the product. And as we have seen with those few brief examples from the European Public Assessment Report, there will always remain a number of residual uncertainties, either purely statistically or purely scientifically, which means that once we have this approved biosimilar onto the market, we definitely will need some more post-marketing data that will be gathered once the product is being implemented for its real use. And with that, I would like to thank you, finish here, and thank you for your